welcome to my video. Today's video is going to be on the topic of the mechanism of antioxidants in the body. Antioxidants have different mechanisms depending on their chemical structure that allow them to work in the human body to reduce oxidative stress. Oxidative stress comes about from free radicals and the damage that they cause to cells in our body. Free radicals are parts of groups of molecules called reactive oxygen species, known as ROS, reactive nitrogen species, abbreviated by RNS, and reactive sulfur species, abbreviated by RSS. Active oxygen species are highly reactive species that can damage cellular components such as lipids, proteins, and DNA. These molecules are produced during cellular metabolism and functional activities and have important roles in cell signaling, gene expression, and ion transportation. However, excessive amounts of reactive oxygen species can have deleterious effects on many molecules, including proteins, lipids, RNA, and DNA, since they are very small and highly reactive. Reactive oxygen species can attack bases in nucleic acids, amino acid side chains in proteins, and double bonds in unsaturated fatty acids, in which the OH radical is the strongest oxidant. Reactive oxygen species attacking macromolecules is often termed as oxidative stress. Cells are normally able to defend themselves against reactive oxygen species damage through the use of intracellular enzymes to keep the homeostasis of reactive oxygen species at a low level. However, during times of environmental stress and cell dysfunction, reactive oxygen species levels can increase dramatically and cause significant cellular damage in the body. Thus, oxidative stress significantly contributes to the pathogenesis of inflammatory disease, cancer, diabetes, Alzheimer's disease, and aging. In order to prevent or reduce the reactive oxygen species-induced oxidative damage, the human body and other organisms have developed an antioxidant defense system that includes metal chelating and free radical scavenging activities to neutralize these radicals after they have formed. Now I'm going to talk about phenolic compounds and the mechanism by which these work as antioxidants. Phenolic compounds have the capacity to act as hydrogen donors or to chelate metal ions such as iron and copper by inhibiting the oxidation of low-density lipoproteins. First, I'm going to talk about metal chelation. Metal chelation is a form of mechanism of antioxidants to reduce oxidative stress. This involves reducing the formation of reactive oxygen species metal chelating ions. Metal chelating usually targets the reduction of previously oxidized ions and their reaction with hydrogen peroxide and the generation of hydroxyl radicals. Metal chelation involves binding to metal ions to form complex structures which are less reactive, and metal chelating antioxidants are considered as secondary antioxidants because they decrease the redox potential and thus stabilize the oxidized metal ions. Phenolic compounds are also able to act as antioxidants by scavenging free radicals. Phenolic compounds have the capacity to act as hydrogen donors, and the mechanism of this is known as hydrogen atom transfer, which is commonly abbreviated as HAT. Phenolic compounds reduce or inhibit free radicals using HAT. Their HAT mechanism involves the transfer of a hydrogen atom from the hydroxyl group to the free radical, which results in the formation of a new radical that is less reactive than the original one. The reaction mechanism of a phenolic compound with a peroxyl radical involves a concerted transfer of the hydrogen cation from the phenol to the radical, forming a transition state of an HO bond with one electron. The new radical can then be stabilized by resonance, which distributes the unpaired electron over the aromatic ring of the phenolic compound. This process makes the new radical less reactive and more stable. The effectiveness of an antioxidant in HAT reactions depends on the strength of the bond between its hydrogen atom and the rest of the molecule, as measured by its bond association energy. Antioxidants with high bond association energies are better able to donate hydrogen atoms to free radicals with lower bond association energies. This is because the energy required to break the bond between the antioxidant and its hydrogen atom is roughly equal to the energy required to form a new bond between the antioxidant radical and the free radical. The hydrogen atom transfer mechanism is particularly important in lipid systems, such as oils and fats, where the free radical chain reactions can occur rapidly. Lipid oxidation can lead to off flavors, odors, and a decrease in nutritional value of food products. Antioxidants can prevent or delay lipid oxidation by donating hydrogen atoms to lipid peroxyl radicals, which are intermediates in the free radical chain reaction. By stabilizing these radicals, antioxidants prevent further chain reactions from occurring and protect the quality and shelf life of food products. In order to design and develop effective antioxidants, Researchers use computational methods to predict the BDEs of different molecules and select the ones with the highest potential for HAT reactions. By understanding the hydrogen atom transfer mechanism and selecting antioxidants with high bond association energies, researchers can develop new and effective antioxidants that can prevent or delay oxidation in food systems. Other types of compounds that undergo hydrogen atom transfer to act as antioxidants are carotenoids and vitamin C. However, carotenoids tend to favour single electron transfer, which we will talk about next. The single electron transfer mechanism is a common way by which antioxidants neutralise free radicals and prevent oxidative damage. This mechanism involves the transfer of a single electron from the antioxidant molecule to the free radical, which stabilises the radical and prevents it from reacting with any other molecules. The single electron transfer mechanism occurs through a series of steps. First, the antioxidant molecule encounters the free radical and orientates itself in such a way that the unpaired electron on the free radical is able to interact with a single electron on the antioxidant molecule. The electron transfer process requires a certain amount of energy, which can be provided by thermal energy or by light energy, depending on the circumstances. If the energy requirement is met, the electron can jump from the antioxidant to the free radical, neutralizing the free radical and creating a new antioxidant radical. The new antioxidant radical is now in a lower energy state than the original antioxidant, and as a result, it may have different chemical and physical properties. This radical may be able to donate an electron to another free radical, or it may be able to react with other molecules in the body. However, because it is less reactive than the original free radical, it is less likely to cause further oxidative damage. Overall, the single electron transfer mechanism is an effective way for antioxidants to neutralise free radicals and prevent oxidative damage in the body. The mechanism is important for maintaining cellular health and is exploited in the development of synthetic antioxidants used in the food and pharmaceutical industries. One advantage of the single electron transfer mechanism is that it can occur rapidly and efficiently, allowing antioxidants to neutralise free radicals before they can cause damage to the cellular components. However, the mechanism can also generate new radicals in the process, which can lead to a chain reaction of oxidative damage if not properly controlled. Therefore, the balance between antioxidant concentration and radical concentration is crucial for effective antioxidant activity. That is everything, and hopefully you've learned something about the mechanism of antioxidants.